Okay, good morning, good afternoon, and good evening. I'm Li Kanliang. So today I'd like to discuss the complexity of generic oligonucleotides, some key challenges in achieving and demonstrating sameness, and also impurity profiles and controls. And finally, I'd like to share with you some of the agency's efforts in addressing these challenges. Here's my disclaimer. The presentation reflects the views of the presenter and should not be construed to represent the FDA's views or policies. Oligonucleotides are short strings of nucleic acids with therapeutic effects. They can be fully synthetic or semi-synthetic, and they can be modified or unmodified. There are many ways to modify oligonucleotides, and modification could be at the 2' position or the 3' position of the ribose ring, or could be on the bases. And the oligonucleotide can be conjugated at the 5' end and the 2' position or the 3' end. The internucleotide linkage can also be modified, and in most cases, this modification is non-stereospecific, therefore leading to 2 to the nth power of diastereomers. This significantly increased the complexity of oligonucleotides. The ribose moiety can also be replaced with a morpholino ring. And the non-stereospecific modification by the PDA or the phosphorodiamidate in those linkages also leads to 2 to the nth power of diastereomers. I apologize for the busy slide here. This table summarizes the 20 oligonucleotides CDER has approved as of July 2024. The last column lists some of the complexity considerations based on the for generic oligonucleotides based on the RLD labeling information. The PS and PDA modification of the linkages actually gives rise to from a few diastereomers to more than 1 billion diastereomers. The same regulatory requirements for generic small molecule drugs also apply to generic oligonucleotides, such as having the same clinical effect or safety profiles and identical active drug ingredients, which translates into active ingredient sameness for complex APIs such as oligonucleotides. Sequence chemical structures, diastereomeric composition, high order structures, and potency and activities, etc., are among the sameness considerations. For most synthetic oligonucleotides, sequences are well defined and controlled and can be verified. Regarding the structures, for the correctness and completeness of the modifications, including the position of the modifications, need to be demonstrated for generic oligonucleotides. The sameness evaluation also includes a physical chemical characterization at the drug substance and drug product levels. Oligonucleotide impurities have some unique safety considerations, such as off-target effects, immunogenicity and inflammatory risks, etc. These risks need to be considered in impurity profile comparison and also impurity controls. It is recognized that binding of oligonucleotides to their targets are largely sequence specific. Then why is diastereomeric composition sameness required for generic oligonucleotides? This is because clinical evidence is not available to confidently negate the impact, and there are also some evidences indicating that stereochemistry and conformation of the oligonucleotides may impact their binding affinity and resistance to nucleus. For example, Iwamoto and others studied myeloimmersion and various stereochemically pure isomers and found that stereo configurations at the PS linkages substantially impact the pharmacological property of the oligonucleotides in their studies. Also, approved oligonucleotides counted all diastereomers as the part of the active ingredient. Therefore, unless adequate data is available to prove that it has no impact, diastereomeric composition sameness should be demonstrated for generic oligonucleotides. The graphs in this slide were adapted with permission from the ACS publication by Russell and others. The authors changed the RNS ratio at some selective internucleotide linkages of the 20-mer by using different activators. Several different methods were used to analyze the samples. Each curve on this slide represents more than 500,000 diastereomers. The first method did not give much information about the composition. Other methods gave, with wider peaks, with some distribution features. Let's look at the MICC method and compare the levels of profile changes from sample 1 to 2, and from sample 2 to 3. The levels of profile change look similar. However, from sample 1 to 2, the RS ratio was changed at only one additional chiral center. But if you look at samples from 2 to 3, the RS ratio was changed at 4 additional chiral centers. This comparison indicates that certain diastereomeric composition changes may not be proportionally reflected in the profile changes by some methods. To address the analytical method limitations, one may consider using multiple analytical methods with orthogonal separation principles that can provide detailed distribution profiles, combined with other orthogonal methods such as an NMR and with additional physicochemical characterizations such as the melting temperature or TM of an appropriate duplexes from the oligonucleotide. The methods should be adequately validated for sensitivity, specificity, and resolving power. If the diastereomer number is low, one may develop methods to separate each diastereomer from other diastereomers and impurities. This way, one can compare the composition directly. To demonstrate sufficient sensitivity and specificity of the methods towards various diastereomeric composition changes, one may consider making multiple suitability test standards for method development and validation. These standards may have RNS ratios changed at a diverse set of a small number of chiral centers. If the methods are sensitive to such diverse and verifiable small changes in diastereomeric compositions, it is more likely that these methods may be able to detect the diastereomeric composition differences between the test product and the RLD. So for distribution profile comparison, one may consider comparing both overall and individual regions of the profiles. The acceptance criteria for sameness comparisons needs to be justified, ensuring comparable safety and efficacy profile as the RLD. The RNS ratio measurement at each of the chiral center after its creation is actually an indirect but effective approach to obtain stereochemistry information of your product, especially when the number of diastereomers is very high. And typical analytical methods may not be able to provide a precise picture of the diastereomeric composition. So it is important for firms to understand the baseline of their product for future potential changes and to ensure manufacturing consistency. This may be applicable to both the new and generic oligonucleotides. Typically, oligonucleotide impurities profiles are very complex with high number of impurities. Some impurities may be due to the incomplete modification or having extra nucleotides or missing nucleotides somewhere in the sequence. The complexity is further compounded by the high number of diastereomers in many cases. The large molecular weight of impurities makes it challenging to separate impurities with small structural differences. The resolving power of a single analytical method may be very limited, and co-erosion of impurities and isomers needs to be addressed. Oligonucleotides are excluded in ICH guidelines for impurities. The FDA has not published a guidance on impurity thresholds for oligonucleotides. And furthermore, the agency has not recommended structure-based classification of oligonucleotide impurities for safety evaluation and quality control purpose for generics. For impurity profile comparison and controls, multiple methods with orthogonal separation principles may be needed to address the co-erosion issues. The nature and location of the structural change in the impurities need to be determined. For example, for inotertion shown in this slide, an impurity with one methyl C unit missing could have 3 different sequences or structures. The exact location of the missing unit may need to be determined unless otherwise justified. If there's any ambiguity, one may want to see if it helps by conducting a comprehensive risk assessment and consider all the possible structures or impurities based on your process understanding. For impurity level controls, RLD comparison is the rule of thumb for generic oligonucleotides. Orthogonal and complementary analytical methods may be used. And when dealing with new impurities, higher impurity levels, proposed impurity thresholds, or impurity grouping approaches comprehensive risk assessment may be needed to evaluate their impact on safety and efficacy. Off-target effect, immunogenicity, and inflammatory risks, and general toxicity, and any other risks associated with the possible impurities may need to be considered. Summation-based grouping acceptance criteria without comparing and controlling the individual impurities levels may be hard to justify for generic oligonucleotides. Some examples including the retention time segment-based grouping, or N-1 or N+1 groupings, etc. Also, the fact that oligonucleotide impurities have higher molecular weights and therefore lower molar amounts than in the small molecule drug case may not be an adequate justification for requesting a higher weight percentage limit in the oligo cases. You may want to see if a comprehensive risk assessment based on the worst case scenarios may help justify some of the grouping approaches. For RLD comparative studies, 3 batches are needed at the drug substance and drug product levels. For evaluating the API from the RLD products, one may obtain the API by an appropriate sample processing method, such as extraction or dialysis, etc., depending on the need and the nature of the RLD comparative study. For diastereomeric composition sameness evaluation, sample processing should not change the diastereomeric composition of the sample itself. If a test is sensitive to formulation or process history, one may consider subjecting both the generic and the RLD samples to the same process. For intrinsic property characterization, formulation impact may need to be eliminated. For example, TM is an intrinsic property of a duplex, but can be impacted by the presence of ions, etc. Therefore, TM measurement may be done at the drug substance level. For siRNAs, TM measurement may be performed at both the drug substance and drug product level to understand the formulation impact and also demonstrate sameness. Sameness evaluation on 3 exhibit batches at release and shelf life, at end of shelf life may be necessary. To support initial ANDA filing in a timely manner, one may consider evaluating a development batch for age considerations and submit the results as additional supporting data, provided that the development batch is equivalent to the exhibit batch in all aspects of CMC. CDER has various programs to facilitate and advance generic drug development, assessment, and approval. Some of the examples are listed here. The agency has published quite a few product-specific guidances on oligonucleotides to help generic product development. The agency also has internal and external research programs to advance the regulatory sciences. Here's an example of the research by our colleagues at the Office of Pharmaceutical Quality Research to address the challenges in analyzing co-eluting isobaric impurities. These impurities were difficult to differentiate and quantify by the conventional methods such as LC-MS. The OPQR work actually illustrated the possibility of quantifying these very challenging isobaric impurities. With that, I'd like to thank all the people and teams and officers listed on this slide. And here are some questions for you to consider. Question 1, OM impurities have much higher molecular weight than impurities of small molecule drugs. And the molar amount of the OM impurities are lower, and therefore OM impurities should generally be given higher weight percentage limit than impurities of small molecules. True or false? So the answer is false. The next question, which of the following statement is true? Oligonucleotide binding is sequence specific. Stereochemistry of the OM modification need not to be considered in sameness evaluation. B, I can use 3 development batches of the generic OM for sameness evaluation and characterization for my submission, even if they are not true representation of the exhibit batches. And C, for complex sameness or comparative impurity profile evaluation, I can use multiple orthogonal analytical methods that gives quite different and detailed distribution curves or profiles on the same sample. Or D, currently I can justify my quality control of impurities in generic OM based on literature information about OM impurity classification without comparing them to the impurity of the RLD. So which one is correct? C, that's right. So to summarize, RLD comparison is the rule of thumb for generic ONs, and when necessary, use multiple orthogonal methods. Also, conduct comprehensive risk assessment to help justify proposed solutions. And as always, seek agencies' help via various channels. And thank you for your attention. Now I hand to Dr. Wang.